Hi, I'm Waleed. I'm a first year medical student at the Albert Einstein College of Medicine. And today, my this project was helped with Dr. Bala from the University of Chicago, Department of Pathology. And they'll be talking about my project, the in silico analysis of differentially expressed genes and prognostic utility in cholangiocarcinoma. So, as y'all know, or may not know, cholangiocarcinoma is unique in that it's a very rare cancer. It's very descriptive or very presentation. What exactly do I mean by that? It's just a very low instance, around zero point 0.5 to around 6 in every 100,000, with some places being above 6 in places such as Thailand and China. That being said, in conjunction with being a very rare disease, in order to the pathology to come across, it's also very varied in how it shows histologically. And because it's not a lot of good prognostic tumor biomarkers, the criteria isn't very detailed in terms of how it can differentiate it early on, and that poses some unique issues regarding CCA diagnosis and treatment. What we should note is that oftentimes it has very generalized symptoms. What I mean by that, you have jaundice, yellowing of the eyes and skin, you have a weight loss, some LFT values are a bit different, but elevated, and these can present as many different things. So these aren't the most useful in terms of really diagnosing a case of CCA. And it's important to diagnose CCA early on because it does have the propensity to metastasize distal lymph nodes relatively quickly with these generalized symptoms, not very established criteria, so on and so forth. What happens is that you don't catch these cases of CCA early on. And by the time you do detect it, oftentimes metastasize. And at this point, surgical intervention is not the most effective, with a 5-year survival rate being around 20%, but I've seen some people showing as low as even 10 or 5%. Accordingly, at this point, chemotherapy is the most effective as well. So in terms of dealing with CCA, even though we haven't advanced in how much we know regarding it, we still haven't correlated that or transferred that into a visible reduction in deadliness in a sense. And accordingly, CCA can actually divide into the three main categories, depending on where you find the tumor itself. If found in the bile ductals, it's called iCCA or intrahepatic CCA, and that's roughly around 20 to 30% of cases. Perihepatic CCA is seen when you have tumors in the right, left, and common hepatic duct, and that's about 50 to 60% of cases. And accordingly, you have distal CCA, which is seen in the common bile duct, and that's seen in about around 30% of all cancers, all cases of CCA. And each of these different subtypes have their own unique, treatment and prognosis and so on and so forth. So we know all this about CCA. What's the point of this project? The point of this project is essentially to find out, using publicly available data sets and online tools, can we sort of make, sort of molecular fingerprint for CCA that going forward might be a useful launching point for further studies into this disease. In order to do so, we used a bunch of different online tools, mainly relegated or mainly looking towards the TCGA or GTEx data sets. These are basically data sets where a bunch of cancers were analyzed, a bunch of mRNA sequences were obtained, expression levels were obtained. And these tools, along with usage of the R statistical package, is what we use to really tease out some useful trends in CCA cases that we observe. So first, we want to see what genes are most often seen in cases with a worse prognosis. Now, to do that, we use something called a GEPIA, or Gene Expression Profiling Interactive Analysis Tool. And we found any gene with a p-value less than 0.01 that was shown to have a correlation with worse outcomes in terms of overall survival was the actual outcome looked at. In case of CCA in the TCGA dataset, we made a list of those genes that were involved. And from those genes, we see beyond just over under expression of those genes, what are any mutations, CNAs, or any genetic abnormality? And to do so, you something called the CBio portal and its relevant tools. Through that, we have this list of genes. We have this list of survival-related differentially expressed genes. Are there any, sort of pathways or biological processes that are heavily found in those genes? What do I mean by that is often when detailing a pathway, let's say you have a pathway, for example, I'm going to talk about a bit more, platelet activation. Let's say there are 80 genes in the entire human body that's really been, annotated for that pathway. If we see 5 or 6 genes in that, previously mentioned list, we might see, oh, this is an enriched pathway. We see these genes often appearing in these biological processes. Maybe some aberration in this process is what leads to, some of the issue with CCA. And that was done mainly through two different tools, CPDB and Metascape. And these are actually portals in which it actually looks at other annotation factors such as KEGS, reactome, bicarta, so on and so forth, in order to give a detailed list, detailed readout. And after that, we have these differentially expressed genes. We want to see what genes are actually related to those genes. Sort of, in a sense, a second order list, if you may. And due to we use Gene Mania, which it's basically an online data set, an online tool that combs your data sets, looking at, co-expression values, observed genetic interaction, even hypothegenetic interactions based on, analysis and computer models, as well as shared domain protein domains, what genes are related to the previously mentioned differentially expressed gene list that we talked about that we got through Zapier. So that's basically, as you can see, there's many different tools. And what's the purpose of this? Again, the main idea is to curate a sort of list of genes that will be of interest going forward in cases of CCA in regards to outcome and survival status. And looking at the results, we did just that. We saw about 70 genes with a p-value of less than 0.01 that was found to be consistently related to worse prognosis, either over or under expression. And you can see here, we then looked into something called OncoKB, as well as looking at the frequency mutation. Basically, beyond just expression level aberrations, did you see any mutations in said genes that might, have some effect in CCA prognosis? As you can see, out of the list of around 70 genes, only about 10ish or so actually had a mutation that was observed in those cases, according to CBioPortal. Again, these are all publicly available data sets. If you have your own private data set, that might show some other issues, and that could be something, again, going forward that we look into when expanding upon this project. And you can see here, we have a variety of genes, all p-values of less than 0.01, some very much less so than 0.01. While the mutation frequency isn't that great, this is about looking on 6 and 7 of the cases, there is the mutation in those genes. And you might ask, what's OncoKB? OncoKB is simply just an annotation portal. What I mean by that is a sense that this is a repository of genes that have been associated with cancers. What has been annotated in cancers. Is it comprehensive? No, it only has about 5 or 6,000 genes annotated already. But the fact that you see some of them not being in OncoKB means that they might not have as much done work on them, and it currently might be of some interest going forward. And again, here are a bunch of other genes that again all just have a p-value less than 0.01 being found in the worst prognosis compared to worst outcome survival. And you can see here is that you didn't see any mutation frequency associated with these based on the 6 or 700 cases that we looked at on the CBioPortal. And going down, this is the sort of gene enrichment / pathway analysis I was talking about earlier. Here we see a lot of it being related to chemical carcinogenesis, and that makes sense given how CCA can develop. Uniquely is platelet activation signaling. I didn't see that much based on previous literature regarding this and how it is to CCA, so that also might be something of interest going forward, along with cellular amide metabolic processes. You see an enrichment, you see an overabundance of genes in this above list regarding the Zapier list found in these biological pathways. So going forward, this might be an area of interest for some researchers to either do more, in silico analysis, in silico, as I mentioned earlier, being basically on computer, or data analysis, trial lab stuff, or even going on to the next stage would be wet lab analysis, doing I'm working with cells, animal models, so on and so forth, to see what the relationship is between these biological processes and these genes and CCA prognosis and progression. Additionally, as to be expected, we see an enrichment of genes involved in nephron development because, again, it makes sense regarding where we're finding these cancers in the body. Not so much as hepatic development, but it might be of note to go forward. And this is figure 2, essentially here. And down here, this is what the Gene Mania was for. This is where we had the aforementioned gene list that expressed, found using Zapier, the one above, shown in table 1. We then ran this analysis and tried to see what sort of genes are also related to these genes. So again, as I mentioned earlier, sort of a second order analysis, a second order gene list of those that might be involved in CCA. And while I didn't put the actual list of the genes down here, there are around 20 to 30. What I did note is that I did put down all different biological factors or biological processes that they're involved in. You see them being transcription factors, involved in the keratinization process, involved in platelet aggregation. These are also, these might be some ways that these aforementioned genes in table 1 kind of have an effect on downstream on CCA prognosis and progression. So again, this is a bunch of different genes that might be of interest going forward. I also conducted some CAM plots and found that a lot of these genes didn't actually have as much of an effect. It didn't have much prognostic utility in other types of cancers, as breast, prostate. I mean that there might be some sort of uniquely CCA related pathway or developmental process that might be of use to clinicians going forward, and how to best characterize CCA, and ideally going forward catch it a bit earlier. So again, in conclusion, with the study, we've observed a multitude of different expressed genes. We got CCA survival with a wide range of biological processes ranging from platelet activation to nephron development. And even those second order genes are involved in a wider range of things, such as, EGFR1, as well as, being involved in RNA transport, which all might give a hint of, how do these genes play a role in CCA development. Additionally, many of these genes aren't the most annotated, as looking at OncoKB as well as a regular PubMed search in regards to cancer. So it might be worthwhile to look at going forward for future projects as well. And additionally, analysis has shown a bunch of different second order genes that might also have an effect on disease course of CCA. And the main purpose of this project was to show that, through this sort of in silico, low cost, very accessible project, you can actually kind of curate a list of genes that might be of interest to do more expensive, more time-intensive wet lab projects and research projects on. And that's basically it for my presentation and for me, Kevin's poster. I would like to take some time to thank Daniel Jacobs and Jessica Juarez for helping me to come up and curate this idea, as well as the proofreading, and of course my mentor, Dr. Bala, for being there for a step away, as well as for financing this project.